My name is Nicholas. I am a teacher at the University of Cape Coast, School of Medical, University of Cape Coast, and I'm the group leader and lab head of the Pharmacogenomics and Genomic Medicine group and lab, also at the School of Medical Sciences. This morning, we are to go through some briefs on genomic data science. And the whole idea is to introduce you to some basic concepts of genomics and practicality in health and disease, and also to try and see if we can let you know a few things about high throughput technologies for data generations. We also look at LC, that has to do with the ethical, legal, and social implications of genomic data usage and all that. I sent you, or I asked Kevin to send you some 3 manuscripts for you to go through so that it will form part of our discussion. I normally like discussing than me doing the talking alone. So I just hope you had the chance to just go through a few of them so that when the discussion comes up, we can have a meaningful discussion. Now, this, I prefer to have discussions in my class. So let's discuss as often as we can. And also let's try and ask questions if we can. Normally, if you ask a question, I would prefer that we discuss it as a class before we eventually come to some form of finality on the question. It's more fun to me than one person doing the talking alone. Multi-omics. Then from there, we'll go ahead and look at high throughput data generation and all those things. Where I'll use one example, but I'll try and give the basic underlying principle behind high throughput screening and all that. So when you look at biological processes, okay, you look at biological processes, there are different individualized levels of biological processes where you may come across the genome, you may come across the proteome, you may come across the RNA collection, metabolome, and all that. So before the concept of multi-omics, we were so much enthused about trying to understand processes that goes on in the genome and the proteome and all the RNA collections, what we call the transcriptome, that was happening. Now, one of the things you realize is that these individualized biological processes provides individual perspective, okay? And they give so much information because if you know so much about what goes on in the genome, then you may be able to make a lot of deductions. If you know what goes on with the total collection of proteins that are available, you'll be able to also do a lot of deductions. But then the challenge that probably comes with it is that, are these working in isolation? These biological processes, are they working in isolation? That's probably the question. So over time, the question of integrated biological systems to help understand how these biological systems interconnect and communicate with each other came in. And that is where the concept of multi-omics started popping up. So although our workshop is saying genomic data science, I'll be chipping in other things so that we appreciate the fact that it is not just about genomics, but it's also about other concepts. I looked at your background and I realized that there are different people with different backgrounds. So I felt that we should try and get to the basics, and I would ask questions as and when I want to ask questions, and I'm hoping that we'll participate. So let's go back to the basics. Let's go back to the basics. Every organism is made up of DNA. And the DNA practically tells who you are, what you are, how you appear as an individual, and the experiences that you will have as an organism. Okay, all right. So we say the DNA is a double helical structure that is made up of nucleotides. Okay, the DNA has basic units that come together to give it what is known as a DNA. Okay, and it is the arrangement of these basic units that practically makes you who you are. Are we okay with that? Good. It is these arrangements that determines whether you are going to be susceptible to a particular disease. Okay, if you get a disease or you get an infection, how your immune system is going to master all its arsenals to be able to counter that infection. When you take a particular medication, it is your DNA makeup that will determine whether eventually you are going to benefit from that drug without any adverse events or experiencing any severe adverse events, or whether you will even eventually get well just from taking that drug. Are you okay with that?
Yes, I will.
All right. Any questions? So for any time I teach, I just, after everything, I just want to ask whether there are any questions before we continue. Any questions, please? Okay, so silence will mean consent then, right? Okay. The basic unit of a DNA, which is known as a nucleotide, okay, is made up of 4 nucleotides that are joined together in what we call a complementary base pairing fashion. Okay, so there are basic rules that govern how these nucleotides eventually get arranged in us to make us who we are. So we say that for the nucleotides, there are 4 of them for a DNA. We have adenine, we have thymine, we have cytosine, and we have guanine. Okay, and these ones have what we call a base pairing mechanism, where an adenine will pair with a thymine, whilst a cytosine will pair with a guanine. So the arrangement of these nucleotides is what eventually makes us who we are. But then, is it just the DNA that eventually translates into who you are as an individual? There is something that is known as the central dogma of molecular biology, which simply says that if we have a DNA, okay, we have a DNA, the DNA would have to be converted under certain processes into an RNA, which eventually also gets processed into a protein, which becomes the functional unit of the DNA. Okay, which becomes the functional unit of the DNA. And the process through which DNA gets converted into an RNA is what is known as transcription. And the process through which RNA gets converted into a protein is known as translation. So the central dogma simply states that DNA makes RNA, whilst RNA makes protein. Are you okay with that?
Yes, I will. Okay.
Okay. So this is just the basics, right? Now, we have a lot of, our bodies are practically made up of DNAs. Our bodies are also practically made up of a lot of RNAs. And again, our bodies are also practically made up of a lot of proteins, a lot of enzymes, and a lot of all that. Okay. How do we describe all these so many things that are practically found in us, what we call the macromolecules of life that are found in us? So the term omics, okay, the term omics practically is looking at the entirety of individual macromolecules. So when we take the DNA, okay, the DNA makes up what we call genes, okay? So if we are able to study all the DNA or the genes in a particular system or organism, then we are looking at what we call the genome, okay? Then we have the RNAs, okay, which from our previous slide, we have said that is resulting from what the DNA being transcribed into RNA. If we are able to get or capture all the RNAs that are in a particular system, then we are looking at what we call the transcriptome. Okay. Likewise, proteins. We get all the protein systems in our body. We are looking at proteome. Okay. Then all the metabolic or metabolites in our system would give us metabolome. Now, with all these things, there is an interaction. So that's where the omics concept came in. There is an interaction that eventually leads to what we call the phenotype. Okay. Those with a biological background, when we say phenotype, how do you understand that?
So phenotype normally talk or looks at the physical expression of an individual or of an organism.
So when you say physical expression, what are you looking at? When you say physical expression, what are you looking at?
So we can look at, let me say, the complexion of the skin, the appearance. So the complexion of the skin is an example of the phenotype.
Okay. So
what can be seen physically?
Good. Let me ask this question then. So if somebody takes a medication, okay, if somebody takes a medication and the person has a stomach upset as a result of the medication, or the person experiences dizziness as a result of the medication, can you classify that as a phenotype of the medication?
Okay, I think I will say yes, because that is what is expressing for us to have, or for us to see that that is the side effect. And once we have been able to identify that that is what, or the effect that the drug is having on the individual, I think we are not wrong if we classify it as such.
But our original definition is saying that the physical appearance, but dizziness and diarrhea are the physical appearances. And another person can answer, please. I want us to just have a fair idea about some of these things, okay? Which would
those are going to be should I say, the symptoms?
The symptoms, okay. Yes, who else? Yes, who else? This class is quite light, so I'm expecting so far we have 38 people online, so I'm expecting a contribution from as many people as possible.
Yes, for me, I can just classify the effect of the medication as the phenotypes because it's a physical expression of the medication that you have taken. Maybe you had there was some there was some interactions with the medication with your DNA, and then it has expressed something like symptoms. So I can say that this is the expression of the phenotype, expression of the medication that you have taken.
Okay. Yes, any other person? So based on probably even just the letter that we've had, okay, if we are asking what a phenotype is, how will you define a phenotype now?
It is the physical appearance. Phenotype is actually the physical appearance of an individual.
But what we just described earlier in terms of the medication and all that, That one, what would be the physical appearance on that one?
Yeah, it might be the harmful effects of the drug. So
Yes.
we can classify. Yeah.
Yes, the harmful effect on the drug, but that is still that's the phenotype that is associated with you taking that drug.
Yes, exactly.
So good. So I want us to capture phenotype so that to capture what would not be more of a physical appearance. So what would be how will you now redefine phenotype? How will you now redefine phenotype?
Sir,
Yes,
I
sir.
think, can you hear me, sir?
Yes, I can hear you.
Okay, I think when we say phenotype, we are looking at the interaction of the genotype and the environment, how the person expresses what is in his gene physically. Yes, the one we see is the one we call the physical presentation is the one we call phenotype. But what that is happening inside is what is the genotype.
Interesting. Okay, so I know you all want to see something physical, but to better capture other forms of phenotypes that practically we do not see, we may add what we call observable characteristics. Are you okay with that? So apart
Yes, sir.
from the physical thing that you see, there are also observable characteristics. So like somebody taking medication and feeling dizzy, somebody eating some food and even having certain kinds of reactions. These are observable characteristics that come with it. So that is another form of phenotype that you will see. So feeding back into where others were moving into the genotype, you realize that if we are able to capture the genome, the transcriptome, the proteome, and all that, okay, we may be able to link some of the genes and variations in some of these genes to certain observable characteristics and maybe physical appearances in terms of phenotype that will make meaning to those genes and variations. Do we get that?
Yes, sir. Yes, sir. Thank you.
So for example, if we say that you are intelligent, right? The only way we can know that you are intelligent is if I interact with you. If I give you a question and you give me a certain kind of answer that I feel, yeah, this is top-notch answer, then maybe I can think from my own opinion that, oh, yeah, this person is intelligent, right? Although intelligence is not a physical appearance, it is an observable characteristic that I saw, okay? So let's say that there is a gene for intelligence, okay? I can only link that gene to intelligence or your level of intelligence based on that observable characteristic, which probably I would properly characterize. Are you okay with that?
Yes, please.
All right. So going forward, the conceptual relationship of the central dogma, okay, was simply trying to capture all molecules of interest in a particular macromolecule comprehensively. So you see that in terms of the genome, we have genomics. In terms of the transcriptome, we have transcriptomics. In terms of proteome, we have proteomics, metabolomics. Okay, we are trying to see if we can capture the entirety of all these macromolecules of interest. Understanding the entirety of genes in an organism is essential for human health, disease, and medicine. Very critical. Your genetic makeup will determine how susceptible you are to certain diseases and conditions. We are Africans, so probably let's discuss this thing before we continue. You remember when coronavirus came, right? the news that was, or the story about Africa was that maybe Africa was practically going to be wiped out because we didn't have the health systems and we didn't have the means to combat corona and all that. Now, from just what I've put across, that your genotype makes who you are. First, how do you think we weathered the storm during the coronavirus pandemic? How do you think we weathered the storm during the coronavirus pandemic?
I've read one article which spoke about the fairly young population.
Okay. The fairly young population, yes. Yes, who else? Yes, who else? How did you weather the storm?
Doc, maybe our genetic makeup also contributed.
Our genetic makeup contributed. Okay.
Yeah, maybe also the environment, you know, the environment we are living in, most of us were exposed to a lot of things.
Exactly.
And as a result, our genetic system is actually maybe improved to some extent to be able to resist some of the diseases, including, if you look at COVID, it was more of flu. And we are in most cases in Africa, we are exposed to flu. And as a result, it's like we are already, even the disease as it was coming, we are already living in the environment that was more familiar. I think that's what I would say.
All right.
I would also say the climatic conditions, mostly Africa, it's warm.
Okay. So you realize that Africa, we say, is a cradle of genetics. So we have a lot of different variations in our genetic makeup. Okay. Our genetic background and our exposures. So as somebody saying, so you look at some of my papers, I say that Africa is a population in health transition. We are already exposed to all sorts of infectious diseases and all sorts of conditions anyway. So our bodies were practically built for these things. Okay. So when Corona came, we already had some form of immunity to fight Corona. So all the predictions about Africa eventually did not stand. Are you okay with that?
Yes, sir.
Yes.
Good. So our genetic background, or who we are genetically, our makeup would influence who eventually you become and the kind of disease, the experiences you have when you take certain medications and all that. All right. So what are some of the methods that are used to study the genome? We have Sanger sequencing, we have microarrays, then we have what we call next generation sequencing. How many of you have used Sanger sequencing before?
So I think it's a normal PCR. Is it?
No, Sanger sequencing is not a normal PCR.
Okay.
I'll just explain later. Yes. So before even I go to this, so when you want to try and understand the genetic variations, normally you may have other methods like PCR, FLP, you may have TACMAN that will help you know simple genetic variations, okay? But if you want to study the entire genome, you may need a little bit of a more complex mechanism because this time you are looking at a lot of genes at a particular point in time, which would mean that if you use the normal PCR, IFLP, TACMAN, you probably spend the rest of your life trying to characterize all the genes in the system. Okay, so Sanger sequencing applies some form of PCR principle. Okay, it applies some form of PCR principle, which employs a base by base sequencing method. Okay. And it's actually this method that was used to deduce the human genome in the famous Human Genome Project. I'm sure you know about the Human Genome Project. In fact, recently there was another thing about the pan genome that came up, right? Are we okay?
Yes, sir, we are following.
Yes. Do you know about the Human Genome Project?
Yeah, but not in detail. I've heard about it, sir.
Okay, what do you know about it?
Okay, what I've heard about it is this is a situation where you see, where you want to understand the background, the genesis of a human being, and also be able to understand what is going on up to at present. Probably, maybe you can also predict the future. Thank you.
All right, all right. So a couple of decades ago, some international consortium came up trying to see if they could sequence the entirety of the human genome. Okay. And that led to the birth of the Human Genome Project. And it is this Sanger sequencing that was used to sequence the whole or the entire human genome. I think the project was completed somewhere around 2003, but I think more work is still being done. Okay. Then we also have what we call the DNA microarrays. Okay. Now, this method is used to determine large number of genes, but then you will need to have a prior knowledge of that gene before you undertake the analysis. Okay. Then we also have what we call the very famous next generation sequencing. Okay, which uses a more complex system to try and help us find what we call the unknown. Okay, the unknown. We'll go a little bit into it when we do the high throughput sequencing. Okay. Transcriptomics is looking at all the RNA transcripts, including those, the coding ones and the non-coding ones. Okay. So when we say RNA, we have portions of RNA, of different RNAs. We have some that will not code for any protein, and we have those that codes for protein. Though they might not be coding for proteins, they influence each other. Okay. So it is important that we get to appreciate the entire RNA transcript in a particular cell so that we will know what eventually leads to a certain kind of protein and protein function happening. Are you okay with that? And practically, when we are looking at the transcriptome, our focus is looking at gene expression. How much of a gene or how much of an RNA is expressed, okay, would tell the kind of protein, the quantity of protein, the quality of protein that is eventually made. Are you okay with that?
Yes.
Good. So example, we have, and we've already established the fact that the central dogma has DNA to RNA and to protein, right? So let's say that you take paracetamol. Okay, you take paracetamol and you need an enzyme A, okay, to break down 400 milligrams of paracetamol. And the volume of enzyme that you ideally need to make or to break down that 400 milligrams of paracetamol is 2 ml. So in terms of the quantity of enzyme you need is 2 ml. If you are going to get that 2 ml of enzyme, which is a protein at any particular point in time, the 2 ml or the quantity of enzyme that is produced is influenced, okay, by the number of RNA transcripts that would eventually be translated into that enzyme A. Okay, so if you have more of expression happening, that means that you will get closer or you'll be able to make your 2 mil. That would eventually be able to break down the 400 milligram of paracetamol. If you make less of that RNA and transcript, then you probably make less of it. Yes, come in. Are you okay with that?
Can you please repeat that?
So okay, so what I'm saying is that you have, I'm sure we've all taken paracetamol before, right?
Yes.
Great. So the dosage of paracetamol is 400 milligrams, for example. Okay, for a normal human being, 400 milligrams, let's say. Okay. When you take a drug, it has to be broken down by an enzyme. And an enzyme is a protein, right? From our basics, this one, okay? This is our enzyme. Okay, so let's say this is our enzyme. How much of this is made will depend on the expression of our RNA. Are we okay with that?
Yes.
Yes.
Good. So what I'm saying is that if let's say that for 400 milligrams of paracetamol, we need, let's say 2 mil of an enzyme A. Okay, 2 mil of an enzyme A to break down 400 milligrams of paracetamol. That means that if you as an individual, if you are able to make the 2 mil, okay, of that enzyme, when you take 400 milligrams of paracetamol, you should be able to break it down normally. Are we okay with that?
Yes, we are.
Good. So what we are saying is that that 2 mil, whether you make it or not, or you make that volume in terms of translation to protein, will depend on the expression of the RNA that you make from the DNA. Are we okay with that?
Yes, makes sense.
Good. And that is where transcriptomics comes in. Okay. So we are looking at the entirety of RNA transcript. However, we also need to see whether this analysis will eventually inform us on gene expression. So for example, at times there are certain people who would have certain diseases. And the first thing that we may want to go and look at eventually is to see whether there is a mutation in that person. Okay. But then you will realize that there will probably be no mutation in the person. The person is coding normal. But when you go to the transcriptomic level, you will see that although the person is coding normal in terms of the gene, there is not enough expression of the gene to be able to make up the protein that could have been used for normal functions. Do you understand that?
Yes,
Are you okay
Prof.
with that? Yes. So when you look at conditions that are related to hemoglobin, something like alpha beta thalassemias, they are more quantitative, okay, where the quantities of the various subunits being made are not up to, which may eventually be influenced by gene expression at the RNA level. Are you okay with that? So some diseases, they happen not because of mutations, but the quantities that needs to be made from RNA level to protein level are just not enough. Are we okay with that?
Makes sense.
Yes, sir.
All right. So how do we determine the transcriptome? Again, we need our microRNA, microarrays which helps us to be able to do that. Okay. Then we have NGS, which can also be used to help us understand the whole RNA transcript that is available. Then we have things like spatial RNA studies. So one of the things that you need to appreciate is that with every passing day, new techniques are being made to try and understand the entirety of our biological systems and our macromolecules. Okay. So over the past several years, we've had things like single cell sequencing, spatial transcriptomics, and these are all forms of techniques that have come up that helps us appreciate and understand entirety of macromolecules and helps us understand the genesis or the etiology of certain diseased conditions. Any questions so far? Any questions so far?
What is the NGS? I think I'm lost on that one.
Okay, so NGS is next generation sequencing. Next generation sequencing, which is, these are complex techniques, okay, that helps us discover unknown sequence information. So for microarrays, for instance, before you undertake a microarray, you should know the gene you are interested in. So let's say that if I have a project I want to look at, and I want to look at the gene expression of maybe the enzyme, and this example, I use it a lot, so don't get offended, okay? The gene that is responsible for madness. Are you okay with that? The gene that is responsible for madness, okay? Before I go looking for that gene, I will probably have a fair idea what gene it is, okay? So if I want to see why we as a class, we are all not mad, okay, although we all carry that gene, I want to look at gene expression. So at the gene expression level, since I know that gene that is responsible for madness, and I have a theory that, but we are all not mad, I may say that, okay, probably the gene is not being expressed enough. So I go and get a microarray kit and use that microarray kit to check the gene expression or how much of that gene is being made, okay, at the RNA level in terms of gene expression, so that we can appreciate why we are all not mad. So in this case, we know. Using next generation sequencing, let us assume we don't know the gene for madness, okay? So it's like a fishing expedition, okay? It's like a fishing expedition. Next generation sequencing helps us go deep into the genome so that we can eventually find genes or a couple of genes that could potentially be responsible for madness. Are you okay with that?
Yeah, I'm okay.
Yes.
Thank
So next generation sequencing is mostly used for more of a fishing expedition, looking for the unknown. Okay. Looking for the unknown. Are you okay with that?
Yes.
So somebody's asking on the platform. Somebody's asking on the platform. So Florencia, can you tell me what you meant by what about the environment?
It was at the first of the presentation when you were talking about just the DNA, and I was expecting to talk something about how the DNA expresses. That's when we talked about the proteome later.
Mm-hmm.
You answered to me.
You said I answered that already.
Yes, thank you.
Okay, I've seen that somebody has also raised their hand.
So I just wanted to ask on the point that you made about when you take paracetamol and then there's an enzyme that would break down the 400 grams. I just wanted to ask a follow-up question on that. Like,
Yes, you can do that.
if, for example, you don't have like sufficient enzymes to break down that, well, can there be a case where you don't have sufficient enzymes to like break down that paracetamol, the paracetamol drug, then leading to it not working?
Okay. So yes, there could be a case where you are not making the enzyme at all. Okay. There could be a case where you are not making that enzyme at all. Then in that case, that means that you will be worth not taking paracetamol at all and maybe going for an alternate medication. Okay.
So there's no way that you can manipulate the DNA to produce that enzyme. You just have to go for another option.
Good. So and that's what actually my research group, the kind of work that my research group does. Okay. So we do a lot of pharmacogenomic work. So in the theoretical way, we may probably want to look at manipulating our DNA, but it will be more expensive to manipulate your DNA than just, instead of going for paracetamol, probably going for brufen or Tylenol or Tramol. You understand that?
Yeah.
Because it will be cheaper than, let me change your medication, than spending thousands of dollars just to make sure that you can code right for that particular enzyme, which will probably be another fishing expedition. You may probably get nothing out of it anyway. It will probably not work. Okay, which will be a gene therapy kind of method. Okay, so one of the things that you realize about, in terms of understanding our genome, transcriptome, and proteome is that at times when we get the answers, we may look for alternatives. So in terms of not making enough enzymes, if a patient comes to the clinic and the patient is prescribed with a medication, and which normally doesn't happen so much in Africa, patients hardly go back to their clinicians to go and report. Okay, patients hardly go back to go and report. I was just telling one of my research assistants about, we are doing some work in hypertension where we are looking at some of the adverse events that patients undergo. And I know patients who have been prescribed antihypertensive medication and they take it and they have adverse events and they say they are not taking it again. Okay, but these patients will not report back to their clinician so that maybe the medication will be changed for them. Okay, so if the patient is not making use of the drug, maybe the person is not making the enzyme that is responsible for breaking that drug down, the alternative would be to change. If the person is not making enough, the alternative would be to modify the dosage. You understand that? So it's based on what we find from the transcriptome, the genome, and potentially the proteome that would help us make a clinical decision. And that is why it is important that we appreciate genomic data science. Are you okay with that?
Yes, it makes sense. Thank you. Can I ask another question?
Please.
Just to follow up again.
No problem.
So with the, you mentioned that in the research that you're doing, that you're trying to see if you can manipulate the DNA. And with that, like, if you do that, then I'd assume there would be dangers in a sense that if you do, then you might be causing another problem, right?
No, we are not manipulating the DNA. We are trying to find out how genetic differences allows people to respond differently to medications. So we are not doing the manipulation. We are just going the natural way where we are profiling patients who have been prescribed with medications. And then we, on interaction with them, they tell us about some of the adverse events they undergo. They tell us about some of the observable characteristics that are out of the normal. So we want to genetically characterize these patients to see whether they are coding normal for the enzymes that are responsible for breaking down these drugs. What kind of food they are taking that could potentially be interacting with those medications. What kind of co-medications are they taking that are interacting with each other? Okay, and all that. So that's what one of the things that we are interested in doing in the pharmacogenomics lab.
Oh, okay.
We are not manipulating.
Yeah, makes sense. Oh, okay. Okay, got it. Thank you.
Yeah. Yeah. And I saw that Rodrigue asked a question. What is the difference between a gene, a DNA, and a genome? I know most of you have different backgrounds. Will somebody want to try this one? Will somebody want to try this one? What is the difference between a gene, a DNA, and a genome? Probably I will do the proper arrangement. DNA, gene, genome. What is DNA? What is gene? What is genome? Would anybody want to try?
Let me attempt, Prof.
Yes, please do that.
I think the DNA is the basic chemical unit of genetic information. But I think in the cell, the DNA are kind of arranged in the form of genes. Then the totality of the genetic of an organism put together is the genome.
So when we're looking at the DNA, we're looking at the basic sequences of a gene, okay? And the gene is literally the functional unit of hereditary. So you inherit some form of genes or some genes from your parents. And the genome is the entirety of genes in an organism. Are you okay with that? It's the entirety of genes in an organism. Are we okay with that?
Yes.
Sorry, Doc, please, can you take it again from the DNA part?
Okay, so the DNA is the basic arrangement, okay, or the DNA is practically the basic arrangement of a gene. And we know that the DNA is made up of different nucleotides that are arranged in different patterns to practically help us become who we are. Are we okay with that?
Yes, please.
Good. Then a gene is the arrangement of this DNA that basically gives us a functional unit of hereditary. Okay, so these genes would act on instructions to make the proteins through the central dogma model. Are we okay with that?
Yes, Doc.
Good. And we are saying that the collection of all genes in an organism makes the genome. Are we okay?
Yes, Doc. Please, you can continue.
So we move from DNA. So the DNA makes up the gene, okay? And the genes make up the genome. Okay, then let's look at proteomics. Let's look at proteomics. Again, proteomics would be looking at the entire set of proteome. Now, again, as we've mentioned, when we are looking at omics, we are looking at entirety, right? So proteomics will look at the entirety of proteins that are present in a cell or a tissue. Okay. Now, when we understand the proteome, it helps us to know the timing and the location of proteins that are expressed. Okay. It helps to be able to follow rates of production, degradation, and even the quantities of proteins at any particular point in time, and appreciate whether there is even movement between proteins and subcellular compartments. Are you okay with that? So capturing the entirety of proteins also answers a lot of our questions. Remember, we've again highlighted the central dogma, DNA, RNA, proteins. What if there's something going on that we're expecting that at the RNA level, there'll be translation to proteins? How do we even know whether eventually the RNA was translated to protein? So that is where proteomics comes in. Over the years, we've come to appreciate the fact that it is not just a full central dogma that actually makes a DNA functional, but even at the RNA level, there could be some level of functionality. So there are RNA molecules that still are able to achieve some functionality at a point in time. Are you okay with that?
Yes.
Okay.
Yes, Prof.
Okay. The difference between appreciating the genome and the transcriptome is that proteomics is a little bit complicated. Okay, proteomics is a little bit complicated because of the fact that we have RNA that eventually gets translated into 20 amino acids, which make up all our proteins, right? In addition to that, proteins can be folded into different ways, can be modified chemically through post-translational modification in different ways, and can also have multiple forms. Are we okay with that?
Yes.
Yeah.
So proteomics is a little bit difficult to work with, although it is still possible. Okay. The other thing is that also because of the complexity of the proteome, reference sequences at times become a little bit challenging. Okay, becomes a little bit challenging. And also the different kinds of methods that are used in trying to get the information out there might not be uniform. However, there are high throughput technologies that have been developed to investigate proteomes, and some include mass spectrometry-based techniques and gel-based techniques. How many of you have come across a mass spec? I think one thing I should have done was to bring a mass spec picture for us to see. How many of us have come across a mass spec? I know you have different backgrounds. Somebody said he has used NGS before. I've forgotten the name. Afonsi, is it Afonsi? Idelfonsi. Idelfonsi, have you used the have you come across a mass spec before?
Prof, I have used UV mass spectrophotometer to determine dye components of Romanowski type stains previously.
Okay. So somebody is asking whether it's possible to make DNA from RNA. Yes, it's called reverse transcription. So William, you can make DNA from RNA. It's called reverse transcription. And that is the principle through which viruses try and operate in us. Okay. And also in the lab, it is a principle through which we are also able to measure RNA transcripts. Okay. It's the principle through which we are able to measure RNA transcripts. When I'm going to do these things, then I'm struggling.
Thank you very much, Doc.
Yep. Okay, I'm trying to do this again. Okay, finally, I managed to do it. Okay, so mass spectrometers do one thing. They measure mass. Are you okay with that? And I'm sure when we say mass, we know what it is. The mathematicians can probably explain better. Okay. So in proteomics, the mass gives information on the identity of a protein. Okay. How that protein is chemically modified and its structure. So when you use a mass spectrometer, what it does is that it tries to measure the mass of the different proteins of interest. And based on the masses, you are able to see the chemical modification and even the structure using other forms of complex methods to be able to determine the protein of interest. Are we okay with that? Whilst differential gel electrophoresis help us know the proteomic profile between cells in different functional states. Okay, that is also another technology that helps us appreciate the entirety of proteins in a particular cell. Are you okay with that? Are you okay with that?
Yes, we are okay.
Okay. So this just to give us a basic idea when we are looking at multi-omics, okay, how we can appreciate it when people are talking about multi-omics, okay. Now, the question is that we're able to get genomic data, we're able to get proteomic data, we're able to get transcriptomic data. What is next? Okay, what is next? So I will just touch on this, but we'll go, when we are doing the high throughput screening, we will also look at some of the base databases that we put some of these genomic data in. But once you get all these things, the information is deposited in databases that helps these people be able to access that data that you have so that they can also try and make sense out of it. Okay, so you don't just get the data and keep it to your chest. You get the data and it has to be put out there. Okay, so this just shows us some of the databases and the kind of content that is normally put into it. Okay, so when you take fibromine, you may get transcriptomic and proteomic data. Okay, if you take dev-omics, you may get transcriptomic data, epigenetic data, DNA methylation, histone modification, proteomic, genomic data. You get all these things there, okay? And the kind of species data that you may get humans, you may get mouse, okay? For some, you may get multi-species, okay? So once we get all these data from the various techniques that we've mentioned, they are deposited in databases. That helps us assess for probably future applications. So in all, genomic data sciences is simply saying that there is a big genomic data ecosystem. I remember I chanced on what Prof. Amega said when he was saying that when we are looking at data sciences, we are practically looking at big data and how we can manipulate. When we talk about big data, what is big data? How big should the data be? Okay, genomic data sciences is simply saying that there is a data ecosystem that is made up of a lot of genomic data that needs to be tapped in to be able to help us appreciate the etiology of diseases and therapeutics. Okay, so it encompasses all the things that the omics system that I've made mention of, in silico technologies, even softwares that you will need to be able to access the data if it's available, the kind of expertise, okay, databases and all that would allow us to be able to appreciate the data that is found in the ecosystem. Are we okay? Are we okay? So it is a very huge ecosystem that has a lot of arms that are feeding into it. So you should either be able to appreciate all these contributors to the genomic data ecosystem. Okay. You cannot be an expert in all, but having a fair idea about these individual contributors would help you to be able to make sense out of it. So for this one, for instance, my expertise may probably be in this part. Okay, I hope you see the arrow, the pointer. This part, okay. Potentially, I'll probably be able to play around this part, maybe play around this part. But some of these things, I'll probably be lost. But there are people, and that's why we have different crops of people who are contributing to this or who are part of this workshop. The mathematicians, the bioinformaticians, they would help us be able to synthesize all these things and make sense out of it. Are we okay with the basics?
Yes, sir.
Yes.
Any questions so far, please, before we go on our first break? Any questions so far before we go on our first break? Yes, I'm inviting questions.
Professor, what is the difference between personalized and precision medicine?
I'll probably call it semantics. Okay, the end goal is the same. Okay, what we are saying is that as an individual, we should be able to take your omics information, okay, and tailor treatments to fit that, including other forms of data that we may have on you. So that's the concept of data science. So if we have your omics information, if we have your other forms of clinical information, we will now be able to create an algorithm that would start you on a medication dosage that will help you experience less or no adverse events, but eventually you will benefit maximally from the medication. So the treatment is tailor-made to you. That is the eventual goal of precision and personalized medicine. Okay.
Okay, thank you.
Yes, any other? Let's go through high throughput techniques in genomics and data science. Okay. High throughput techniques in genomics and data science. I'll try and just go through as fast as possible so that we can discuss the ethical LC, the one that I sent you in the manuscript. So when we are looking at genomic data science, okay, when we're looking at genomic data science, we want to appreciate how computational and statistical methods are used to decode information that are hidden in the genome, okay, which, as we've already mentioned in our previous lecture, can be downgraded to genes, which can then be downgraded into DNA sequences. Okay. One of the things you realize is that high throughput techniques have increased and so many methods have been brought out. However, in as much as a lot of data is being generated, being able to decode the information that is generated from high throughput techniques is where the challenge comes in. Okay, and that is where data science is very important. Okay, because what is the essence of generating the data if you can't make sense out of the data? Okay, it is like having money in the bank and you can't spend the money. Okay, so if somebody gives you $1 billion and yet you can see you have $1 billion in your account, and yet you can't spend the money. Then what is the essence of having the money? The same way, we have a lot of genomic data that is available from the different techniques that have been made. But then, trying to decode or decipher information from this data is where we have a problem. Then the other thing also has to do with the fact that even in situations where we have the means to decode this data, what are the ethical, legal, and social implications and responsibilities that should be associated with genomic data, as the data that concerns a particular individual has ethical, privacy, and identity issues associated with it? Do you understand what I'm saying? So the whole concept, the bigger picture, is to see if we can consider all these points in the discussion that we are going to have. Are we okay?
Yes, sir.
Yes.
Okay.
Yes.
So remember when we started the previous one, I made mention of the ways of, so this one, we are zooming into genomics. We may probably not focus so much on the proteomics and the transcriptomics, but mention it one or the other. Great. Is that there were several methods of getting genomic data, where I said Sanger sequencing, microarray, and next generation sequencing. Now, when we are looking at high throughput technologies, okay, we're looking at using complex methods to be able to get as much data as possible from a particular organism. Okay. And so when genomics started, the methods that were known, eventually based on our understanding of the central dogma, ended up with PCR and RFLP that were used to try and elucidate genetic variations, gene expression, and all that goes on in an organism. So my PhD supervisor will tell me that when he started, when he was doing his PhD, now we have thermocyclists that can do PCR. But for them, you literally have to have a lot of water baths where you set them at specific temperatures so that you move from one water bath to the other manually to go and undertake PCR. Now we have PCR machines or thermocyclists that we need to just put in our cocktail and put it in, set our reaction times and various cycles, and we just go sit somewhere and then hope that we'll get a product out of it. And if everything is right, you get a product out of it. Then you're able to go to subsequent downstream applications. You see, so we have traveled far. We have traveled far as a race and as a generation. Generating genomic data has actually moved and improved over the years. Okay, so right from PCR, RFLP, we had what we call the first generation sequencing technologies, which practically was made up of the Sanger sequencing, which I told you was the one that was used to decipher or used for the Human Genome Project. And as at that time, it could cost $3 billion to be able to use Sanger sequencing to sequence a particular gene. Okay, so the Human Genome Project cost so much money because they were using Sanger sequencing. Then over the years, we had what we call the second generation sequences, okay, which had to do with things like the 4-5 sequencing, 454 sequencing, which from Roche We have the several generations of Illumina, several generations of ion torrents that came up. Okay. And because it became more available, cost also came down. Okay, cost also came down from between $5,000 to $10,000. Okay, to sequence a gene. Now we are moving to the third generation sequencing technologies, which has to do with complex Illumina systems, nanopore sequences, and all that. That now costs as low as $1,000 per sequence. And it is believed that there will be another generation of sequences where it will be so cheap that you can now even sequence a gene for just $10. So you see how far we have traveled and why now we have so much genomic data available, and yet we are struggling to even decode the data that is available. Okay. If you have a question, please just raise your hand and talk, because I normally going back into the chat and getting rid of it, I struggle a little bit. Okay. Okay. So why the need for all these several generations of sequences? Okay. Several. Simple is good. Simple is very good. Okay, simple is very good. Remember, I've just mentioned that my PhD supervisor mentioned that when he was doing his PhD, they literally had to carry Eppendorf tubes from one water bath to the other. I'm sure you know what you all know what water baths are, right? We know what a water bath is, right? Water bath normally used at the lab.
Yes, please.
Okay, so he had to do that. This was a simple method of doing PCR-FLP, which could still give the result that was needed. But then it's very complicated, because that means that you have to make sure that your temperature in the water bath is set right, and that no power fluctuation will change the temperature at any point in time so that you can get your product. Okay. However, this low throughput era, where we have to use these simple methods, helped us understood the basics of genomics. Okay, until the first generation sequences came in. Okay, and this is where we have got into. So let's take a quick look at the first generation sequencing that had to do with Sanger sequencing. And Sanger sequencing just makes use of understanding the structure, okay, understanding the structure of the various nucleotides. You should remember the nucleotides, right? You still remember the nucleotides?
Yes.
Good. So when we are looking at the nucleotides, okay, it is made up of a base, a ribose sugar, and a phosphate backbone. Okay. And for RNA, the sugar is called a ribose. For DNA, the sugar component is called a deoxyribose, which simply means that an OH has been taken off from position 2 of the structure and replaced with an H. Okay. When DNA is being synthesized and extended, okay, through replication, what you realize is that the new nucleotide that is supposed to be added to the structure, okay, gets added at the 3 carbon position of the ribose sugar. Are you please seeing the arrow?
Yes, it's visible.
Okay, so it is added at this point. Okay, so in replication, you realize that the nucleotides are added at this position. Now, until something prevents addition of a new nucleotide, okay, it will continue till we get to the point where there is nothing to add. Okay. So for Sanger sequencing, the principle is that we would block the 3' position, okay, of the sugar where addition is supposed to happen, okay, so that nothing can be added. So the OH is removed, and that is what gives rise to the D-deoxyribose, okay? And then the H is labeled with a fluorescent molecule, okay? The H is labeled with a fluorescent molecule, which can then be read, okay, using a detector. Are you okay with that?
Yes.
So for a Sanger sequencing, we have our cocktail, okay, of a DNA template. Then we have the DD dinucleotide phosphate. So remember that the nucleotides are individualized, right? So we have A, T, C, and G. Okay, so you have DDA TP, DDTTP, DDGTP, DDCTP. Okay, then we have polymerase, we have primers, we have normal DNTPs that are thrown into the cocktail. And as the process of synthesizing DNA in vitro goes on, the DD dinucleotide phosphates, which have been modified to have fluorescent molecules, would be incorporated into the various DNA strand, okay, which eventually gets decoded and reconstructed so that you can identify it as a fluorogram or an electrogram using different colors. That would then tell you the sequence of DNA that is found in that particular gene. Are we okay? This is how Sanger sequencing works. Are you okay with that?
Yes, yes.
Yes, sir.
Okay. So it is, and this as described simple, it's not as simple as it is. Okay. This will take several hours of preparation. You do PCR, you do cleanup, you go through a lot of processes before you are able to sequence one gene of interest. Okay. And I'm telling you that this was the method that was used to decode the entire human genome probably 3 decades ago. Okay, 2-3 decades ago. So you can imagine how laborious this is. Okay, how laborious this is. And this method, what it does is that it has a maximum read, okay, of just about 800 base pairs. So that means that for several genes, you would have to do several of these things to be able to get it right. Okay. So that is where the next generation of sequencers came in, where high throughput technologies came in, where you employ these technologies to try and quantify or locate majority of genes that are harbored in a particular cell. Okay. And high throughput technology helps describe transcription, regulation, and mutations that can occur across the entire genome of an organism. Across the entire genome of an organism. And one of the things you realize is that we can do this in less hours and at a cheaper cost. Okay, in less hours and at a cheaper cost. So Gonzinski et al., for instance, in 2002, sequenced a human genome in just 5 hours and 2 minutes. And if and they used high throughput sequencing. If they were using Sanger sequencing, I'm sure that was going to take forever to do that. Okay. And as I've already mentioned, different types of high throughput technologies have been made. Okay. Examples include the ion torrent, Illumina, and nanopore sequences that are now available in different sizes, different versions, different models that can be employed based on how much money you have, the sample that you have, and what you are interested in measuring or quantifying. Any questions so far?
Professor, what is the difference between Sanger sequencing and the normal PCR that we are conducting? Is there any difference between the two?
What? Please ask the question again.
I'm asking what is the difference between Sanger sequencing and the normal PCR that we are doing?
Oh, normal PCR and Sanger sequencing. So the normal PCR, there are no DDNTPs, one. And I've explained to you what the DDNTPs are. I'm saying that for the DDNTPs, okay, or deoxynucleotides, we have blocked the 3' and it's from the DNA. Remember DNA already, we have already taken one OH group. Okay, that is why it is deoxyribose. Okay, so we take another OH from the 3' carbon end and we replace it with an H that has a fluorescent molecule attached. Are we okay with that?
Yeah, okay.
But the normal PCR, we only have the NTPs. Okay, so you see the image that I have here? Okay, so the normal PCR will have the template DNA and have polymerase, the NTPs and primers together with other few things, but it will not have this DDNTPs, DDTTPs, DDGTP, and DDCTP. Okay, so the normal PCR doesn't include all these fluoro-modified deoxynucleotides. Nucleotides. Do you get that?
Yes, actually, these fluorescent molecules. These must be the that we are using, for example, what we are using. I forgot the name. Cyber green. Yeah, these are cyber green, right?
No. So these are fluorescent, specially tagged.
Actually,
okay, so that the detector can detect it. Cyber green is another form of detection of things, but this Sanger does not use cyber green per se.
Okay.
Okay. So in gene expression profiling, for instance, we may use cyber green so that our thermal cycler will be able to detect the cyber green, which is then used to tell us the number of times a gene is expressed.
Okay. Yes.
But this one, because of the specialized nature of it, it is tagged in a way that the equipment can detect it. So it is a proprietary reaction.
Okay, Professor. Thank you.
Any other questions, please? Any other questions, please? I'm trying to go to the chat box. Maybe there's a question there. Okay, so there's no question. Any other questions, please? Okay. If there's no question, then I can continue. So the question then is, what are we targeting in high throughput sequencing or using high throughput technologies? We may target the whole genome, we may target the whole exome, or we may have what we call targeted deep sequencing. Who can tell us about whole genome sequencing? Who can tell us about whole genome sequencing? Yes. Somebody said he has used NGS before. So who can tell us about whole genome sequencing?
That's the full genome of a people.
Pardon?
So the genome sequencing is the full genome of someone. So is it computer to determine maybe the DNA?
Mm-hmm.
So
Okay.
that's like your single mark.
Mm-hmm. Okay. Yes. Is this Sodi? Sodi, you said you have used it before.
Yeah.
Yeah.
Yes. What did you use the NGS for?
Yeah, I use the NGS data, which is constituted of what we call a BAM file and I don't know, single nucleotide polymorphism.
Okay.
SNP.
Okay. Okay.
Yeah.
So when we are looking at whole genome, again, we are looking at the entirety of DNA. Okay. That makes up a particular gene of interest. Okay. Now, when we talk about whole exome, so when you take a particular transcript, remember the central dogma again. When DNA is moved to RNA, the RNA that is made, okay, is made up of what we call introns and exons, okay? And it is believed that the introns are the coding regions that eventually gets translated into functional proteins, okay? But there is a process that happens called splicing that allows these introns to be taken out so that the exons can join. So whole exome sequencing, simply what it does is that it is looking at the coding portions of RNA. That is the portions of the RNA that eventually gets translated into a protein of interest. So next generation sequencing can just be processed to look at that. Do you understand that bit? Do you understand that?
Yes.
So just look at the image. So this is what we have: red, green, red, yellow. So whole genome sequencing is looking at trying to get information on, yes, come in. Whole genome sequencing is just trying to look at information on the entire genome. So we have our gene 1, gene 2, gene 3. We have all these spaces that are made up of introns, okay? So whole genome sequencing does not really care whether these are introns or they are exons. We are interested in everything. Are we okay with that? Whilst whole exome sequencing is interested in the coding viable regions of the genome. So it is interested in just the red, the green, and the yellow, which are supposed to be the genes that are of interest to us. Are we okay with that?
Yes.
Good. Then we have what we call targeted deep sequencing. Okay. What this one does is that there are two methods. We have what we call the amplicon-based enrichment and the capture-based enrichment. Okay. And what the amplicon-based enrichment does, okay, is that you just design primers, okay, that will just amplify the region of interest. So let us say that we are interested in gene 1, okay, we are interested in gene 1. Our primers would just target gene 1 only, okay, and gene 3 only. It is going to leave out gene 2, so that eventually we will just amplify gene 1 and gene 3. Are we okay with that? Whilst capture-based enrichment, on the other hand, what it does is that it will fragment the DNA first before we enrich and amplify the region of interest. Okay, and the method you use to get your data, again, would depend on your starting material and how rich you are, and the kind of resources you have to be able to analyze the data. Are you okay with that? Are you okay with that?
Yes.
Good. So recently, my group and some collaborators, we submitted a grant, and because of the money that they were supposed to offer, it was just about £750,000. And our plan included utilizing whole exome and whole genome sequencing. So the plan was to use a low pass sequencing method before we go deep into it. And one of the reviewers wanted to ask the reason behind it. And we said that, but the money you are giving us is so small. And looking at what we wanted to do, it would be difficult for us to go deep into whole genome sequencing because that one is more expensive. So the money you have would influence the kind of technology and how deep you try and go into. If you don't have a lot of money, you don't go targeting the ones that involve a lot of money, okay? And the data that can be generated from this high throughput technologies includes metagenomic data, transcriptomic data, ribosomic data, that's where you also get to know more about proteins, epigenetics, which I haven't mentioned yet, whole genome sequencing, which can be for either humans, bacteria, viral, depending on the starting material and where you got your source from. Who knows anything about epigenetics? Who knows anything about epigenetics?
I think, can I go on?
Yes, please, go on.
Yes, I think epigenetics has to do with what regulates the expression of a gene vis-a-vis whether that portion in the genome or that portion where that gene is in the genome is exposed for transcription or not. So there are some molecules and the way the DNA is folded, when it is or some methylation and some other things, which determines whether they are available for transcription or not. And that determines whether that gene will be expressed or not.
Okay. So what happens in epigenetics is that there are modifications that happen on the DNA sequence that do not necessarily affect the nucleotide itself. So the modifications actually happens on the individual nucleotide, but it doesn't change the nucleotide as in the case that happens in a mutation. And as he said, this eventually goes ahead to affect transcription and gene expression. Okay, so next generation sequencing can help us appreciate whether the phenotype we are seeing is happening as a result of gene expression that is affected through epigenetic modifications or mutations, or whether there is any other thing that is happening. This Sanger sequencing will not be able to help us do it. Normal PCR might not be able to help us do it. Okay, so these are some of the applications and probably the interesting bits about high throughput technologies that are available that helps us appreciate genomic data sciences. Okay. However, high throughput comes with its own challenges. It answers a lot of questions. Okay, it answers a lot of questions. However, there are varying degrees of maturity, technical limitations, and they are not noise-free. So what that simply means is that if you want to answer a particular question, you should be able to choose the right high throughput technology to be able to produce the data of interest. And if the data is available, you should know which high throughput technology was used in generating that data and what statistical and analytical method you will need to be able to analyze that data and how far the answer to your question would go. Are we okay with that?
Yes.
Okay. But these challenges are good. Okay. We still manage to find a way of producing enough data that can still be applied in data science. Okay. So in high throughput technology data generation, how is the data generated? How is the data processed? How is it validated? How is it stored? And eventually, how is it utilized? You are going to get big data, but in genomic data science practice, all these points that we've listed, how is that one going to influence the utilization of that data? Okay, how is that one going to influence the utilization of that data? One, your starting material. Your starting material. You will need to extract your genetic material of interest from your cell. And once you extract your genetic material, you will need to undergo an enrichment protocol, okay, where you, whatever interest you have in any portion of the gene, you enrich that portion before eventually you go ahead and run a quantification step, okay, to be able to determine your interest in that particular gene of interest. Are you okay with that? And this method runs through all high throughput sequencing technologies. Are we okay with that? So the steps are common. We extract, we enrich, we quantify, and then we do our analysis. Are we okay with that?
Yes.
Are we okay with that? Any questions so far?
Professor, what is the meaning of enrichment? How
So
can
when
we
are looking-
enrich the fragmented material?
Okay, so when we are looking at enrichment, all that we are saying is that the part where we are interested in, okay, we need to try and make as much, so we want to make it distinct. Are we okay with that? We want to make it distinct. Okay.
Okay.
Remember, it's an entire genome. We might not be particularly interested in every aspect of the genome, but the portion of the genome that we are interested in, we try and enrich it, okay, by doing things or using methods that will let that bit stand out so that we can get the information that we want to the best of our abilities. So for instance, what I showed here, okay, you see that, are you seeing the arrow? Are you seeing
Yes,
the arrow?
Professor, I can see.
Okay.
Yeah.
So you see that we have gene 1, gene 2, gene 3, right? We are interested in gene 1 and gene 3. So what we do is that we enrich it by trying to use methods that will enhance only gene 1 and gene 3. You see that? So that when
Yes, I
we go
can see.
ahead with our analysis, the data we get would be for only gene 1 and gene 3. Gene 2 is depressed. Are you okay with that?
Yes, Professor. So for this, we will design the primer.
Pardon?
For example, if we are interested in some specific genes, so for these, we will design the primers, specific primers.
Exactly. So this one, for instance, if this is a amplicon-based enrichment, so if you want to do amplicon-based enrichment, then you will need to design primers that will flag the gene of interest. So you see the arrow there. So we design primers that will just be interested in gene 1 here and gene 3 here. So once we toss the primers in, the primers will just bind to the genes of interest. Okay, then we use our enrichment protocol to enrich the genes of interest. Are we okay with that?
Yes, Professor, I got it. Thank you.
All right. Any other questions, please? Any other questions, please? Can I continue?
Yes, please proceed. Yes.
All right.
Yes, proceed.
So this is just to let us see the different phases of high throughput technology or sequencing platforms that have been made over the years. Okay, that have been made over the years. And there are several of these platforms, as I mentioned in the other slide. We have the Illumina, we have Ion Torrent, we have 454, we have the Pack Bio, we have Nanopore Technologies. I've provided a website, so you can always go and have a look at how it is. And again, the choice of this platform will depend on what you are interested in and how much money you have. So in my lab, it's not a very big lab. We have managed to acquire one of the nanopore sequences that we are going to be using for our sequencing research. Okay, but one of the common and popular platforms that have been utilized in next generation sequencing is the Illumina platform. So we'll just go through the Illumina platform. However, as I said, the principles are the same. These are the basic steps practically in all the sequencing processes: extract, enrich, quantify. Okay. Okay. So let's look at Illumina. Illumina is a very common platform that is used and has been used in generating a lot of genomic data. Okay. It's been used in cancer research, microbiology, agri-genomics, complex diseases, reproductive health, genetic and rare diseases, and different kinds of, data from Illumina. Okay, different kinds of data from Illumina. And one of the things you realize is that, again, over the years, okay, different versions of Illumina platforms have been made. And the choice of which platform you want to use, again, would depend on the data that you want and your availability of funds. Okay, so here you see the common applications for the various platforms. We have ones that can look at large whole genome sequencing, small whole genome sequencing. So the large whole genome sequencing will be looking at either humans, plant, or animals. The small whole genome will be looking at microbes and viruses, other bacteria, and other things. Exome and large panel sequences. Targeted gene sequences, that's amplicon-based, the one that I showed you. Single cell, transcriptomics, target gene profiling, microRNA, RNA analysis, DNA protein interaction, ChIP-seq, and all that. But you see that each of these equipments, okay, cannot just go ahead and measure everything. So if you go and use ISeq 100, it can only look at small whole genome sequencing, targeted gene sequencing, target gene expression profiling, and the microRNA and small RNA analysis. Okay, but if you come to something like the NextSeq 1000 and 2000, it can literally give you data on everything that you want. So if you have money, then you are going for this one. Are you okay with that?
Yes, sir.
Good. And there is another form. Illumina has really progressed. Okay, they have what we call another form of NextSeq series. Okay, that can further do things that probably we cannot all imagine. Okay, but again, it is about the resources you have at any point in time. I remember when I relocated to Ghana, I think about 4 or 5 years ago, I wanted to do one of my works, as I said, my lab is the pharmacogenomic and genomic medicine lab. So we're doing this cardiovascular study where we wanted to look at some whole exome sequencing. So I went to one of our popular research facilities here, and they had the MySeq, which is an Illumina platform. Then I went to ask them, oh, please, how much will it cost if I want to sequence one gene? Then the guy just looked at my face and told me that, oh, it's going to cost $1,000 per sample. So I asked the guy, what is your basis for calculating that $1,000? Because I from where I come from, I could do this for less than $100. So why are you trying to charge me $1,000? He couldn't give me any answer anyway, but eventually the bottom line is I didn't go. I didn't go because I felt that it was a ripoff. The same $1,000 can probably analyze 10 samples for me. So I'd rather stay put and look at a platform where I can use utilize my money than that place. Okay. But each of these series has a certain amount of data that can be generated at any point in time. So if you are using the iSeq 100, which is one of the lowest platforms, you may probably generate 1.2 gigabytes of data within 19 hours. Okay. The MiniSeq, MySeq, NestSeq 550, NestSeq 2000. So you see, the NestSeq will give you like 330 gigabytes of data within 48 hours. So you can imagine if within 2 days, you can have this amount of data, then try and imagine in a year, the amount of data that you can make from this particular machine. Do you understand what I'm saying?
I have a question, please.
Yes, sir.
Yes, please, you can ask.
Yes. So when you say the depth of, when you say the amount of data that can be generated from this different content, does it mean that for maybe if I have an organism now and probably the size of the genome is maybe 1 million base, for example. So is it that it's going to generate a deeper depth of data if I use MIC, next generation 550? Is it that it's going to give me greater depth, or is it going to be multiple of the same thing? I don't understand what it meant by when you say this big amount of data that is being outputted. What would be the use to me if it's still the
it is
same?
going to give you a deeper depth of data on that gene of interest. Okay, so remember, look at the number of reads per run. So you look at the iSeq, it gives you 4 million reads per run. And look at the NestSeq, 1.1 billion reads per run. Okay, 1.1 billion reads will capture more data within a particular gene of interest compared to 4 million reads. Are you okay with that?
Yes, I'm okay. Thank you very much, sir.
Good. Because if you go on the reads per run within a particular gene, the data that you get, okay, would be an accurate representation, okay, compared to when there are less reads. Are you okay?
Yes, perfectly okay. Thank you, sir.
All right. Any other questions, please?
Yeah, my question is, so through all these platforms that you are presenting, so if a researcher is interested, for instance, in a specific population in Ghana, so can he get the data he needs for his research through those platforms?
Yes, you can get the data. Two options. Either somebody has already done it and deposited the data somewhere, which will get to that page, or if you are going to start from scratch, okay, you are going to start from scratch, you need to look at the amount that you have, the resources that are available, okay, the technologies that are available within your reach, not just within Ghana, but outside your reach, that you can use to get that data you are interested in. Are you okay with that? So for those of you that work with or have worked with or worked in labs or supervisors that have funds, at times you see that you go and present a proposal, oh, Prof, I want us to do this and this. Then they will tell you that, well, we don't have enough money to do this, but maybe if you can consider this, it will be fine. Okay. It is not like your idea is not fine. It is brilliant, but the money that is available, that idea would literally spend all that money. So let's go for a less expensive option that can still help us generate a certain level of data or a certain amount of data that will also answer our research question. Zachary, do you get it?
Yes, yes, thank you.
The challenge that we have, okay, there was something I wanted to add to this slide, but I decided not to because I thought it was going to be too controversial. Okay, is the fact that in our African sub-region, most of the genomic data that is generated, unfortunately, is done outside of Africa because we do not have the funds for it. So most of the collaborations that we have, normally, it is about you sending your samples outside. So we literally do the recruitment, collect the clinical data and phenotyping data and everything. Then we send everything out to our partners out there, who then do all the wonderful high throughput sequencing and analysis. So they practically keep the data because they have the means in terms of financial capacity. Okay, that has been unfortunately the challenge we have in Africa. We do not have so much funding for some of these extraordinarily high throughput sequencing. So except a few countries that have high tech research facilities, most data that is generated is mostly from even Africa, data on Africa is generated from outside Africa. Are we okay?
Yes, sir.
Yes.
All right. Yeah. So Illumina allows what we call parallel sequencing of the nucleic acids, okay, which I explained in the question that the person has, whether we have deep and all those things. So the number of times that you go over the gene of interest will tell you the kind of data that is what literally generated, okay. This is practically the workflow of the Illumina platform, okay, the workflow of the Illumina platform, where you do your nucleic acid extraction, you do your library, what we call the library preparation, then you do your sequencing, and when you are done, you do your data analysis. The most critical part of getting high throughput data, okay, is your starting material. Is your starting material, which is the nucleic acid of interest. Why do you think that is so? Why do you think that is so? Why is our starting material of critical interest in our work or in data generation?
Hello.
Yes, I'm listening, please. Or we are listening.
I would say this is more like garbage in, garbage out. So if what you are starting with is not of high quality, certainly you are going to have results that are inaccurate. So it is important that whatever materials that we need to start the work should be of high quality, so that at the end of the day, it will affect the results that we are going to, it will affect the accuracy of the results that we are going to get at the end.
So garbage in, garbage out. It's as simple as it is. The quality of our nucleic acid, okay, should be enough to help us with our library preparation. It should be free of compounds that can inhibit enzymes that will be used during the library preparation. And also, we should make sure that we extract enough genomic DNA or RNA that is good quality to help us get the data that we require. Are you okay with that? And to do this, we either use UV spec, fluorometric assays, or gel-based assays to be able to help us see whether our starting material is of good quality. So for instance, we have these what we call nanodrops that can tell us whether our DNA is of good quality. We have qubits that can also tell us, and we can run a simple gel with a gel electrophoretic and apparatus that can tell us whether the DNA we have is of good quality. And then that will tell you whether you can go ahead and go to the next step or not. Are you okay with that? Are we okay?
Yes, we are okay. Yes.
All right.
Yes,
All right.
yes, we are okay.
Okay. So once we've managed to extract our DNA, the next step is to go ahead and do our library preparation. Okay, our library preparation. And this is the part where we are going to fragment our DNA, which means we are literally chopping our DNA into smaller pieces, okay, so that they can be sequenced in a parallel fashion. Are we okay? So the range of the fragment sizes will depend on the sequencer you are using and the sequencer application. Are we okay with that?
Yes.
Good. Then once you fragment your DNA, you do what we call adapter ligations, which are oligonucleotides, okay? They are also what I will call synthesized or artificial nucleotides, okay? That will be used to prime our nucleic acid fragments. Are you okay with that? So you see here that these are fragments, right? Then there are adapters that are added to our fragments, okay? So these are artificial oligonucleotides synthesized that are chemically added to our fragments, okay, to enable sequences to appear. Okay, and these adapters are sequencer specific. So the adapters that you find in ion torrent sequencing will be different from what we find in Illumina sequencing. So you cannot use adapters or reagents for Illumina sequencing for ion torrent sequencing. Are we okay with that?
Yes, sir.
Okay. Then once you are done with your adapter ligation, what you now do is that you do your quantitation. Okay. So you quantify your library and see whether the quantity of fragments that you have, okay, is at an optimal concentration to be sequenced in the sequencer, okay? And all these things will be done, okay, in the machine. Once you are done, then we go to the next step, which is the sequencing, okay? Sequencing, clonal amplification and sequencing, where you now, so the sequencing is exactly like what we have done in the Saga sequencing, just that this time we are doing parallel sequencing, remember, okay? So our DNTPs are labeled again, okay, with fluorophore molecules. Okay, with fluorophore molecules, which as they are being captured on the platform, it will be read. Okay, and the sequences are provided in this format. Are we okay with that? So you would have several of these reads based on the number of times the fragmented DNA is read. Are we cool with that? Are we okay with that?
Yes, please.
Okay. This is just a general explanation of how we obtain our data, okay, genomic data. And as I said, I gave you a basic run through of what happens, okay. Ion torrent, whatever they practically go through, the only thing that happens, the difference will happen in the reagents, the library preparation and all that. But eventually, we get our data in formats that can be analyzed, okay. Now, we have our data. I see that there's one of us who has done some bioinformatics. So that is good. And the person is also in my university. The person is also in my university. Once the data is generated, what's next? The data that is generated is not like, so for here, you see that it is so simplified, right? We are looking at ACA, ATC, GAT is for our explanation. But what comes out is not as simple as it is, okay? So the data needs to be processed. It needs to be analyzed. Then it needs to be cleaned, okay? So that eventually we can interpret it in a way that would make sense after all. Are we okay with that?
Yes, sir.
Good. So processing our data. What are we looking at? Remember, we had adapters, we had other things that were done. So eventually they will all come in with all sorts of interference and noise at the background. Okay. So what we do is that we try and see how we can clean the data up. We can try and see if we can look at some variant calling and gene expression and then evaluate in terms of what we call annotation. Okay. So for cleaning up data, what we are looking at is that we are doing what we call base calling. And we know the bases, right? The nucleotide bases, A, T, G, C, right? We did that in our first lecture, okay? So processing would require us being able to base call, being able to determine the number of reads and length of sequencing, and then filtering and trimming adapter sequences. Then what we realize is that in some of the sequencing platforms, at times we do what we call multiplexing of samples. Okay, multiplexing of samples. So what we do is that if, for instance, you do not have enough flow cells to work with, at times you can tag each individual sample, okay, with certain adapters and pull all of them together and then do the sequencing. Once you are done, you undertake what we call demultiplexing of samples. So now you separate the individual samples, okay? And all this happens in the processing part of the data analysis of your sequencing data, okay? Then once you are done, you do your analysis where you map to reference sequences, you move artifacts, you look at annotation, detect variations, and also try and see if you can determine gene counts, okay? Gene counts. Then once you are done, what is the eventual meaning of the data that you have obtained? Because if I realize that I've discovered a gene, but then what is the relevance of that gene? Okay, so you have a car. You know this is a car. But then if you don't know the relevance of that car, then why do you need, or why do you even need to have that car? You understand what I'm saying? Are we on the same page?
Yes.
Yes,
Good.
Professor.
So we have,
following.
but I'm listening. We have our data now. We have managed to do our analysis and done all the reads. Okay, and we found something. The question is that, how do we interpret this and predict gene functions and biological relevance? Remember, one of the concepts of data science is to be able to do prediction, right? One of the concepts of data science is to be able to do prediction, right?
Yes, that is it.
Exactly. So now we have our genomic data. So in terms of prediction, how are we going to do that? Okay, we should be able to look at the biological and gene relevance in terms of function of whatever discoveries that we have made. Okay, whatever discoveries that we have made. This is just to kind of tell us the different platforms that I've mentioned and the kind of amount of data that we can make at any point in time. Okay, and this is, again, all that I've said, how we can get our data eventually. Okay, now let's look at what we call the integrative approach. The integrative approach. Remember, we are looking at multi-omics, right? We are looking at multi-omics. We have our data. Okay, we have our data. The question is that, is genomic data in isolation? Can anyone answer that? Do you think the genomic data that we obtain, it is in isolation?
Yes, Professor.
Why do you think it's in isolation?
Because you have too many steps from the bigger to the little information.
All right, but then what about the central dogma then? Will the information on the DNA alone be able to help us appreciate the biological significance of whatever discoveries in the gene that we have made or the transcriptome that we have made?
So the integration of different tools, as you said.
So the beautiful thing about this is that we will need an integrated approach to be able to go through the whole spectrum, okay, of the central dogma to see whether there is any biological significance to our discovery at the genomic level. Okay, and that is where bioinformatics comes in. Okay, so we have our genomic data. We will need a platform that will help us appreciate what happens at the transcriptome level, what happens at the proteome level, and using all these bioinformatics platforms will require tools, and we tapping into databases that would eventually help us get the required results. Do you understand that? Do you understand that?
Yes.
Okay.
Sure.
Now, I gave you, I made Kevin send you a paper on statistical analysis of next generation sequencing data, a partial overview. Now, the question is that, based on whatever thing you read on that paper, what do you think, okay, what do you think influences your choice of bioinformatic tools and biostatistical tools? That is this going to form part of our discussion. Then from there, we'll go ahead and then we'll do the LC. What do you think would influence your choice of bioinformatics and biostatistical tools and analysis?
Of course, the research question, and also how much money do you have, and the availability of data. So there are many factors that influence.
Okay, yes, who else? What would influence your choice of bioinformatics and analysis? I actually send these papers for these sessions of the workshop so that we can discuss briefly. We will not go into details. We just discuss briefly. What will influence your choice of bioinformatic tools and biostatistical tools?
Maybe the type of information that you have.
The type of information that you have. Okay. What else? What else? Relevant knowledge on the biological sample to understand the bioinformatic tools. Okay. Yes, what else?
What about the outputs that you want to generate?
The output you want to generate, okay? The output you want to generate, okay? What else? All the answers are in the manuscript I sent. That's why I sent them, so that you can read and have a fair idea. The required results, yes, what is your study objective? Very important, because if you are interested in looking at gene expression, you cannot go and use tools that will only give you variations. So yes, your study objectives will influence that. Yes, what else? The kind of data that is available would influence your choice of bioinformatics tools and analysis. If you have data that is going to help you understand genetic variations, then you will need to use tools that will help you know those genetic variations. You need to tap into databases that will help you understand the biological relevance of those variations. Okay. What about the equipment used? Do you think the equipment used would also influence the kind of bioinformatic tool and biostats that you employ? Do you think the equipment that you used or the platform that you use will influence that, and why?
I'm thinking, as we referred to in the few moments back, in the last session, we are looking at the amount of generated and amount of information generated and quality improves with the better equipment.
So the equipment used may also influence your choice. So one of the things you realize is that some of these platforms, they already have their bioinformatic pipelines that you can use, while some may have open systems where you can tap on any other bioinformatic pipelines to use. Okay, so the equipment you use or the platform that you use in generating your data would also influence the bioinformatic and biostatistical analysis that you employ. For those of you that haven't read the manuscript that I sent, please go ahead and read it. You appreciate what I'm talking about. Okay, it's very important. Now, when this data is generated, they are deposited. Okay, they are deposited in databases that you and I, if we are interested in looking at or making use of that big data, we can go on and try and get access to that data and use. So it is not every time that you have to go through the processes that I have mentioned earlier on in generating the data. At times, somebody does what I call the donkey work. Then you continue that donkey work by trying to utilize the data that has been generated and deposited. Do you understand that? So you don't need to go through all the processes that I've mentioned to generate that data. But what you now need to do is that, how do I get access to the data and utilize? Do you understand that?
Yes.
Good. So these are some of the databases which I pointed out in the previous, that you can deposit data in, okay, and utilize, okay? Then what I've done is that I've just brought you some useful websites that you can learn next generation data analysis on. So this is a part that I said Prof. Chimusa was supposed to help you do that, but he had a clash of activity, so he couldn't, okay? So you can just go to these sites and it will help you play around some data, and you can learn basic next generation sequencing data analysis, which eventually maybe in future we can build on for our use. Are you okay with that?
Yes.
Yes. Okay, I'm not sure if I think the recordings are made available. I should think so. The recordings are made available.
Yes.
So if they are made available,
Yes.
then you can just go Google these things and you'll find them. Are we okay? Okay. So once the data is generated, they are deposited into what we call databases. Okay. And these databases are designated databases for future use. So when I say future use, future can be tomorrow. Okay. So I do the donkey work in my lab. I generate all the data. And once I am done, I need to deposit it in a designated database so that tomorrow somebody can go and tap into that database and use. Last month, I wanted to publish a paper in the BMC Genomic Medicine. And this is a microarray data that I did on a drug metabolizing enzyme. So when I submitted the paper to the journal said that I need to deposit it in a database. And I really struggled to get that into a database. Eventually, I managed to get into a database and then provided a session number so that anybody in future who wants to go make use of that data can go tap into that microarray database and use it for whatever thing that the person wants to use it for. Okay. Are we okay with that? And these databases, what they do is that they contain information on nucleic acids, protein sequences, structures, genomics, single nucleotide polymorphisms, mutations, other forms of mutations, and copy number variations. Are we okay with that? And examples of some of these databases for genomic data is highlighted here, or they are highlighted here. Okay, so if you want access to any genomic data, you may go onto any of these sites and you will be able to have information on certain genes of interest. As long as the information is deposited there, hopefully you should be able to have information on it. Are we okay?
Yes, sir.
Okay. So now we get back to another tricky part. So we have generated our data, right? We have deposited it in the database. The question is that who gets access to our data? Is it that anybody can just go, I sit in my office or I sit in my lab and I tell myself that, oh, you know what? I think I need the genomic data on all Ghanaians who have diabetes. So let me look for a database and just go download that data and just use for my pleasure. Okay. Getting access to databases would depend on how the repository is managed. So one, there are some databases that has an open or unrestricted access policy, which simply means that anybody can just go there and download that data and use. The only caveat is that if you are downloading that data, you are expected to use the data set responsibly. Okay. Then there are databases that have registered access, which simply means you need to register so that they can vet your credentials before they will allow you to use that data. Okay. Then we have those that have controlled access. Okay. You need to request a particular data set from them. Okay. So recently I wanted data on health and demographic systems, which is hosted by USAID. And I had to literally complete a form and write an abstract. I give them a project title of what I wanted to do. And then they asked me, which data are you interested in? I have to literally tick the data I'm interested in before they gave me access to just those portions. Then I realized that, oh, actually, I couldn't get the clinical data. So I sent them another message. Then they asked me, I should justify why they should release the clinical data to me. And I had to justify. So this is a part where you need to register for access to the data, one. Then even you need to make special requests to get access to that data. Then there are databases that are what we call mixed access. So depending on what you want, you may get access to certain portions of the data, whilst on certain portions of the data, you need to make special requests before you are given. Okay. Are we okay with that?
Yes, sir.
I have a question.
Yes, please ask.
Do you have to pay to get in access?
This is an interesting question. I wanted to bring in this bit, but let's go to it. That's fine. So the data sets that are available, some of them, ideally, you don't have to pay. But until recently, we have what we call private databases. So this data is being hosted by private individuals. Okay. And once they are being hosted by private individuals, there is cost involved in managing this data. So for you to get access to this data, you may have to pay for it. But if your data is found in a public database, you wouldn't have to pay for it. So most of the funding agencies would require you to deposit whatever sequence data you have in a public database so that it will have free access. Okay. But if you have your own money and you generate your own data, you may decide to put it in a private database, which then will mean that anybody who wants access to that data will have to pay for it. Are we okay with that?
Yes, thank you.
All right. Yes, any other question? Any other question before we continue, please? Any questions? Because I want us to look at the other two papers that I sent, which has to do with the ethical, legal, social implications in omics data and usage. So we've seen the different databases, how we can get access to them and all that. Based on the manuscript I sent you, the one that looks at ethical considerations in research with genomic data, then ethical concerns on sharing genomic data, including patients, family members. And this question is up for discussion, so it's not a trick question. We are discussing based on the paper that we had. If your own genome data is out there, how will you want it to be assessed and used? How will you want it to be assessed and used? Yes, this is an open discussion. It's open to the class.
Considering the implications and the opportunities, I think in a very ethical, private manner, I think where it's safeguarded, I think that would be the most optimal. Considering the kind of information that can be generated from it, I think it's reasonable to have safety mechanisms in its access.
So if, let's say, you had your genomic data out there, would you just want anybody have access to it and using it to the person's satisfaction?
Not necessarily. I would prefer where within the certain confines that data can be accessed, where there is assured security of ethical utilization of that data. Then I think I wouldn't want just anyone to access it.
Okay, yes, who else?
Can I speak?
Yes, please, you can.
Sir, I think so whenever we are sending our data to the organization, they already have the policy. So on the basis of policies, we will able to send our data, and the policy is already having some important instructions to use the databases. So I think so. This is the way how they are dealing or giving the access to the other people.
Yes, who else? If your data were out there, would you really want it? Anybody just getting access, and why?
Prof, I think for me, I would be very comfortable that if it's my data, if I give consent to it, I would be comfortable that that person can use my data. Otherwise, without my consent, I would not be comfortable that someone should have access to my data. And also how that data should be used. Of course, if I've given consent to it, it should be used according to the agreed within the realms of the agreement, not beyond what we have not agreed.
So that brings us to another concept: consent. Consent. Do you think people stick to consent as agreed on? I don't know how far you're going to research, but do you think people stick to consent as they have agreed on?
In most cases, I think, Prof, people tend to go outside the agreed terms of reference. And I think it's unfortunate, but there are some maybe that would follow what has been agreed, but some would maybe go a little bit more beyond what was agreed.
Because in our system, how many people follow up? You know, when you sign an informed consent, normally there are parts that tells you that you should be, anytime you are not interested in the research, you can call and they should discard your samples. I don't know how many of you have used informed consent, right?
Yes,
Do we have,
Yes,
and your data needs to be destroyed. Do you think these things happen? Do you think these things happen?
No, really no.
Why? Who is supposed to ensure that they happen?
The researchers or the research center, maybe, but I think that it doesn't happen.
I think the ethical review boards also have a hand in these kind of things. So like when someone asks to have their data removed, there has to be like the follow-up processes with ethics review boards to ensure that people actually, the researchers actually, they keep to that end and they make sure that they mention that someone wanted their data removed and that it happened. I think.
Yes, I agree.
So if you're talking, we can't hear you.
Teacher, we don't hear you.
Yeah, so I'm saying that IRBs have a role to play in protecting the privacy and interest of research participants then. Right?
Yes.
Because if IRBs are granting ethical approvals for studies to be done, then they have to ensure that the study or the data that is generated is utilized strictly according to ethical standards. Right?
Yes.
Right?
Right.
Okay. Then the question then is that, what about incidental findings? So let us say that you gave your consent that they should find out whether you carry a gene that will let you be susceptible for diabetes. Then in so doing, it was realized that you also carry a gene that will let you be susceptible to, let's say, hypertension. Do the researchers have the right to inform you, or you don't want to know? And why?
Yes, because it's an ethical aspect to inform maybe that possibility for my health. I think that is part of this kind of research.
So you want to know, or you want them to inform you?
Yes, they must inform me.
But what if they don't? What if they don't inform you? Can you take them on?
I'm not sure.
What if they don't inform you? Especially when the data has been deposited in the database.
I'm not so familiar with the consent procedures, but I'd assume that when consent is given, such scenarios would be included to say that this is what we're doing and these are the things that potentially might come up. So in that, when that happens, I'm thinking that you would be discussing that with the participant. Because I think that the researcher would know the possible potential things that could happen in the study.
But I also think that the researchers are like, they usually do these kinds of studies for their own benefit, right? So sometimes when it comes to incidental findings, I don't think they would always let the other person know that, look here, this is what we found, especially not in the consent form or like part of the study that they've consented to.
Okay.
I think that is one of the benefits the participant gets from the research. So he or she is supposed to know whether he know his status or something of that sort. So it's a benefit the participant derives from the research.
Okay.
Thank you.
Okay. All right. So I mentioned private database. Yes.
I would say, as a participant, how would you even know, for instance, that the researcher is withholding this information from you?
That is where the ethical and legal implications come in. How would you even know? Yes, Adam, your hand is up.
Yes, so my contribution to this discussion will be that, one, there should be a policy on, or a system for you to be able to, one, deposit your data. If, let's say, we are doing something on so your genomic data, there should be a policy or a system that these are the procedures that, one, you upload the data there, and then the ethical issues that must be considered when any researcher want to use such an information. And we know that in research, there's this issue about confidentiality and not using, is it privacy, and not using direct patient identifiers. So if we're able to have such a policy which will address all these issues in using human participant or subject for your research, yes, at the end of the day, we are going to have, a more principled or more ethical way of accessing some of this data. And then before you can even get access, you should apply to the body that is hosting the data, and then we'll go through. So once you apply, then that means you go through the process, the processes that are required, like a proposal for experts and people in are also into ethics look at your proposal to see, one, what you are using the data for, and then two, what are even some of the harms, anticipated harms, and then the benefits that at the end of the day your study is going to, or we are going to derive from your study. So once all these things are met, I think we are going to have a more principled or ethical way of accessing genomic data.
Can I speak?
Yes, please, you can.
So in my point of view, when I was conducting my study, at that point I have collected the data from my participants. There, firstly, I have take the consent from my participants and tell them like why I'm doing this study and what would be the purpose. So on this basis, I collect the data and then I analyzed and work on it. So I just do in this. I just done my research work in this way. So they will. So in this way, I just getting the access of the data. Because whenever we are getting the access of the data, we have to tell to the participants what we are doing. So I think so. This is the way how we can going through the process.
Okay. Yes. Any other contributors, please? Any other contributors? Any contributors? Now, I mentioned private databases, right? Would you want to send your data to such a private database? If yes, why? If no, why?
Yeah, I think if it is highly regulated and there is a laid down procedure that you are sure of, that at the end of the day, they are adhering to good ethical principles, I think it shouldn't be wrong to access or to use such a private databases.
Okay. Yes. Would you send your data to a private database? Would you want to do that? Would you want to do that, and why?
Maybe I will want because I'm thinking in the future if I need it for some new illness for me or my family, maybe. But as a research, I prefer in a public data or a private database.
Yes. Last contributors before we try and run down.
Did I miss the question? Which one are we looking at?
We're looking at the private databases.
Okay, the private database depositing data.
Yes.
Okay. I'm thinking the potential benefits of this they can actually be very valuable. Of course, with the ethical access, they can be valuable. Let's say in looking at their possibilities, we can actually detect early occurrence of diseases. If that can actually happen by accessing the DNA, the information for me and my family, then I would say it's valuable as long as there is ethical utilization of that information.
Interesting. Interesting. Okay. We've spoken about anonymity and all that, right? When we are saying that we are each unique for our DNA, or are we not?
Yes, we are.
Good. Then if we are each unique for our DNA, can DNA information ever be truly anonymous?
It won't. I don't think you can have anonymity with the DNA information.
You don't think so? Why?
Why? My reason here is if every fingerprint is unique, there are not two people with the same fingerprint. So it's possible to track my identity using my fingerprint. So it is the same with DNA. If it is unique and there is possibility of tracking an individual using their DNA, so you can't really say it's anonymous.
Agreed, Prof. A paper spoke about the increase in DNA repositories, and also if you have a family member, who may have their DNA somewhere else, it's very easy to trace back to you. So DNA can really never be anonymous.
We can always backtrace. If you get somebody who is interested in finding out something, the person can eventually work things backwards and identify the person.
Correct. Yes.
Right. So how do we then, when we say anonymity, what are we then trying to say then? What are we trying to say then? What are we trying to say then? All that we are looking at is at least trying to take our personal identifiers, right? So maybe let's make it difficult for the person to actually identify us. What do you think? let's make it difficult for the person to identify us, right? At least the person shouldn't have it easy, right?
Yes.
Because if your name is linked to it, then the person can easily identify it, but at least it might probably take a couple number of years to identify you after all.
So you definitely have to remove any kind of identifying demographic characteristics, as you said, name, address, and so forth, and hold that in a separate file and just link it by a unique coded number or something like that.
Exactly. Exactly. Yes. Exactly. So we need to make sure that we clear these what I call personal identifiers, which will make it difficult for people to identify individuals. However, accessing genomic data, especially in the public databases, it's not a very difficult procedure, and that is because of data sharing principles, which helps us to see how best we can all find what I call a better world for ourselves, okay? Because knowing the genomic composition in a certain gene, in a certain population might influence discoveries of certain medications and understanding certain disease susceptibilities and mechanisms and pathophysiologies that would help us stop the progression of that disease. Remember when COVID came, the first group of people that sequenced the coronavirus were very hesitant to release the data. I don't know how many of you were following it, maybe because of my interest, but they were quite hesitant to release until probably there was a lot of bashing going on before they released the first set. Then suddenly coronavirus data started coming out. Then we started looking, understanding the rate at which it was getting mutated and new variants were coming literally every day, right? New variants were coming every day. Eventually, we came to understand the coronavirus more. Vaccines were produced and things started happening. And now the World Health Organization has declared coronavirus as a case of non-emergency. So now you can live your normal life. All restrictions are thrown out of the window. You just do your best to make sure you don't catch it anywhere along the line. Anywhere along the line. And just, it's like you are now up to yourself. We are not making any rules. Just protect yourself. You get that? But what I'm saying is that data sharing is a part of us. Okay. And I gave my own example of me trying to publish the pharmacogenomic data that I was told to deposit the data in a database so that people can have access to it. Okay. Although I created my own conditions, I told them that, well, I haven't published all the data, so I'll publish it in maybe 2-3 years time. So the data is there, but for people to have access, I request that they hold on to the data for the next 3 years. So they replied and told me that the very moment I publish that data, they are releasing it. And I've already sent one out for publication. So now I'm worried that maybe they'll be able to put the rest out there. You get that? So those are some of the things. But again, a lot of policy. Yes, please.
Yeah, I think with what you said, depositing, okay, as part of the discussion, depositing your data in a database, I think it is also to improve transparency in research in general. Yeah, so if, let's say, I have a genomic data and then we are not sure of the source, how I even came by the data, and I publish a paper out of the data I have, and I indicate based on my research, these are the things that I found. How sure can we be that indeed you even collected the data in the first place? You didn't sit, let's say, in your lab somewhere or your room somewhere and generated the data to look, more perfect. So depositing it allows other researchers who are also interested in that area to request, once the person goes through the normal or the procedure that is required to access your data, then they can also see if they can reproduce or replicate your results and then say that, yes, indeed, the findings that you got from your study is a reflection of what you found or of what the data you got is saying.
Yes, yes, that is also one aspect of it for publication purposes, for verification and probably validation and all that, which is, for me, it's also quite important that we get to appreciate getting access to that data. It's and using the data and somebody confirming whether what you found is actually true or not true. Although again, there are always caveats that come with it. Okay, there are always caveats that come with it. But I think the eventual story is that there are a lot of genomic data that is out there in the ecosystem. And we need to appreciate that there are thousands of analysis that we can do to be able to answer health issues. But we need to make sure that we work on these things in a responsible manner. Okay, in a responsible manner, that will be good for what I call humanity. At least if for nothing at all, I think coronavirus really taught us a lesson. Coronavirus really taught us a lesson. And so we should always be open when it gets to accessing and utilizing genomic data, but then in a very responsible fashion. Are you okay with that?
Yes.
Okay.
Yes, yes,
Any,
yes, yes.
Any questions, please? Any questions?
Yeah, I have a question.
Yes, please.
So you're talking about the COVID-19 related
Please,
data, right?
can you, I think, I don't know, I can't hear you properly.
Right now, can you hear me?
Yes, can you increase the volume a little bit?
Okay.
Uh-huh.
Okay, Professor, right now, can you
Yes,
hear me?
yeah, we can hear.
Okay. So I want to ask, like most of the laboratories, they having already the data related to the different diseases. For example, I'm talking about one of the, you can say one for example, I have the sample related to the appendix. Okay, I just send that sample to the laboratory to get the results. So that means they have recorded that data, whatever they are getting the results, right? So how we can collect or getting that data of the different patients from the clinical from laboratories who already having that databases. Do we pass from the same process? Like we have to give them the, you can say we have to show the consents and then getting the data.
Okay, so let me just recap and make sure I got your question right. You are saying that maybe you may have some data that is available and you want to get access to that data for certain studies. And do we have to go through the same process to be able to get access to that data? Getting consent maybe from the original patients and all that. Is that what you are saying?
Yes.
Okay. So let's say in a clinical setting, okay, in a clinical setting, normally when you collect patient data and it's kept in a repository, when I, as a researcher, I'm interested in that data, I will normally apply through the IRB that functions in the system, where they will consider what I'm using that data for and everything, and decide to agree or not disagree to me getting access to that data. Do you get that?
Yes. Yes,
Good.
yes, I got it.
So I don't necessarily have to go to the patients, okay, But now currently we have a situation like that where my student has collected some data, but then I'm thinking of calling the patients to ask them certain questions. And I will need to go back and ask them of the ethical implications. That do I have a right to call the patient? It is not any invasive question. I just want to ask them that the medications they are taking, are they having any adverse events so that I can catalog the adverse events. But I don't want to call the patient, how did you get my number? You get that? How in God's name did you get my number? Okay. So the IRB will give you approval once they realize that your protocols are right and they don't break any ethical protocols. Okay. You may not have to go back to the patient. Like I have data for 7,000 patients. I can't call all the 7,000 patients and see contents. Some of them are probably dead anyway, or maybe they've traveled. And so even if I call them, I'll not get them. You get that?
Yeah.
Exactly.
Yeah, got it.
Exactly. Yeah.
Okay, thank you so much.
All right. Any other questions, please? Any other questions, please? Any questions? All right. So thank you very much for your time and for the wonderful discussion we've had today. I hope that at least this would just provide a basis for you to understand what genomic data science is about, how the data is generated, the ethical implications, and the relevance of knowing what tools that you require to be able to analyze that data. My email address is there. Anytime you have any questions, you can please send me an email and I'll do my best to answer it to the best of my ability. Is that okay?
Yeah, got it.
All right. All right. And if I hear of any bioinformatic workshop that is, in fact, there's actually one that is coming up now. It's being organized by H3 Africa, H3 Bionet. Okay, H3 Bionet. I've typed it in the console, H3 Bionet. Just go to H3 Bionet and then see what they are doing there. There's a current workshop on bioinformatics that is coming up. Okay, H3 Bionet. That will help you be able to understand what is going on. And maybe you'll be able to learn some 1 or 2 bioinformatic tools from there. Okay. Are we okay?
Yeah, please.
Yeah,
All right.
got it.
Yes, thank you.
All right. So
Thank
thank
you
you
so
very
much.
much. And I hope to talk to you sometime soon.